Welcome to the deep dive. Today, we're diving into something that honestly sounds straight out of science fiction. This whole idea of young blood being able to rejuvenate older tissues. You've probably seen the headlines, right? Studies showing, really amazing effects on the brain, blood vessels, even muscles. It's a pretty captivating thought, isn't it?
It really is. The potential seems enormous.
But here's the twist we're exploring today, and it's a fascinating one.
Hmm.
What if this system that actually carries all those potential rejuvenating factors?
Mm-hmm.
What if it doesn't really respond?
You mean the blood system itself?
Exactly. What if the delivery system is like immune to its own medicine? So today we're digging into some really important research. It's a 2021 paper called Aged Hematopoietic Stem Cells Are Refractory to Blood-Borne Systemic Rejuvenation Interventions by Theodore Ho and his team.
A key study in the field, yeah.
Our mission is to unpack what they found, pull out the key insights for you, because it really throws a curveball into how we think about aging. So I guess the big question is, why? Why would the blood system, especially these critical stem cells, be so resistant to the good stuff it seems to deliver everywhere else?
That is precisely the puzzle. And to get it, we really need to talk about these hematopoietic stem cells, HSCs for short.
Right, the source of it all.
Pretty much. Think of them as the master cells, the absolute blueprint for your entire blood supply. They make everything red cells, white cells, immune cells your whole life.
So absolutely crucial for staying healthy.
Totally fundamental. Without them working properly, well, things start to go wrong.
And I assume, like everything else, they age. What happens then?
Oh, they definitely age. It's a significant decline, actually. As we get older, these HSCs, they lose function. They don't work as well.
And that has real-world consequences for people.
Absolutely. It's linked to really common age-related problems. Things like anemia, feeling tired and weak because of fewer red blood cells, or a weaker immune system, which means getting sick more easily.
Makes sense.
And even a higher risk of certain blood cancers. It's a big deal.
So what causes this decline? Is it wear and tear or something else?
It seems to be a mix of things. Some problems are cell intrinsic, meaning they happen inside the HSC itself, like DNA damage building up over time, or maybe the cell's copying machinery getting stressed, metabolic changes.
Internal problems.
Exactly. And then there are cell extrinsic factors. That's more about the neighborhood the cell lives in, the bone marrow environment or niche that changes with age too, becomes less supportive.
Okay, so internal issues and external environment problems.
Right. Both contribute to HSC aging.
So given all the excitement about young blood fixing other aging tissues, the logical next step was to think, hey, maybe it can fix these aging HSCs too. That must have been the hope.
It absolutely was the hope. It made perfect sense on paper. If you see rejuvenation in the brain and muscles, why wouldn't the source of the blood itself benefit?
Yeah.
But this study really challenged that assumption dramatically.
Okay, so how do they test it? How do you actually expose old blood stem cells to young blood in a controlled way?
They used a technique called heterochronic parabiosis. It sounds complicated, but the idea is fairly simple, if a bit Frankensteinish. They surgically join a young mouse and an old mouse, so they end up sharing the same blood circulation.
Wow. They literally share bloodstreams.
Exactly, for about a month in this study. And this technique, parabiosis, it has a track record. It has shown really strong rejuvenation effects in other tissues in previous studies. Brain, muscle, liver, quite consistently.
So a powerful tool. They point this rejuvenation machine at the blood system itself. What did they find? Did the old blood stem cells get younger?
And here's the kicker. No. They found basically no observable rejuvenating effects on the old HSCs.
Seriously, after a month bathed in young blood.
Seriously. The old HSCs, they kept showing the typical signs of aging. They still had high levels of this marker, CD150. You can think of it like a little flag indicating the cell is aged. Their ability to regenerate the blood system, their actual function, was still reduced. And they kept producing the wrong balance of immune cells, that myeloid bias we mentioned, which is bad for immunity.
So the young blood just didn't touch them.
Didn't seem to. And interestingly, the flip side was also true. The young HSCs, even though they were exposed to old blood factors, they weren't harmed. They stayed young and functional.
That is striking. Such a contrast to the brain or muscles. Did they double check this? Maybe parabiosis itself has weird effects.
Good question. They did check. They also did experiments just injecting plasma, the liquid part of blood, from young mice into old mice.
Same result.
Same result for the HSCs. No rejuvenation. What's really convincing is that in those same mice getting the young plasma, they did see rejuvenation effects in the brain.
Wow. Okay, so the plasma was having systemic effects, just not on the blood stem cells.
Exactly. It really isolates the resistance to the HSCs themselves.
So, if the fountain of youth, this young blood, isn't working from the outside, what's going on inside these old HSCs? Did they look at the molecular level?
They did. They went deep into the cell intrinsic stuff. Even after the old HSCs had been swimming in young blood factors via parabiosis, they still showed the molecular scars of aging.
Like what specifically?
Well, they found these things called nucleolar gamma H2AX foci. That's basically a molecular signal flare saying there's ongoing stress, particularly when the cell tries to copy its DNA replication stress.
So the internal machinery is still struggling.
Right. And consistent with that, they saw lower levels of proteins needed for DNA replication, like MYC-MT4, and the cells actually divided more slowly. They were sluggish at a fundamental level.
Okay, what else?
They also looked at metabolism. Old HSCs had problems with their energy production, deregulated oxidative metabolism, and lower mitochondrial membrane potential. The powerhouses of the cell, the mitochondria, weren't working optimally.
And the young blood didn't fix any of that internal stuff?
Nope. All those intrinsic problems just persisted.
What about the neighborhood? The bone marrow niche? Did the young blood at least spruce that up for the old cells?
Another good question. But no, not really. The aged bone marrow environment, which is known to be less supportive, for instance, it loses certain cells called osteoprogenitors. It didn't improve with the young blood exposure either. And those pro-inflammatory signals, cytokines, that are often elevated in aged tissues, they stayed high in the old mice's bone marrow.
So neither the cells nor their immediate environment got rejuvenated by the shared young blood.
Pretty much. It really points towards the problem being deeply rooted within the old HSCs themselves.
This is kind of a big deal. If young blood doesn't work, what about other things? You know, the classic anti-aging strategies we hear about all the time. Did they test those on HSCs?
They did. And the results were just as surprising, frankly. Let's take exercise.
Okay, exercise is good for everything, right?
Generally, yes. It's known to help rejuvenate brain stem cells, improve cognition, lots of benefits. But in this study, exercise did not improve the function of old HSCs or fix the aged bone marrow niche.
Wow. So old mice that were runners still had old-acting blood stem cells.
That's what they found. The HSCs seemed resistant to the benefits of exercise that other tissues clearly get.
Okay, my mind is slightly blown. What about the other big one? Calorie restriction. Eating less to live longer.
Right, calorie restriction, or CR. It's famous for extending lifespan and health span in many, many studies. Some earlier work hinted it might help HSCs in middle-aged mice.
What?
But in this study, looking at properly old mice 24 months old, which is quite aged for a mouse lifelong calorie restriction, showed no improvement in HSC function.
No improvement at all.
None. Their HSCs still had high CD150, poor regeneration, the myeloid bias, the age niche. CR didn't seem to touch the core HSC aging issues in these old animals.
This really hammers home that different parts of us age differently, doesn't it?
It absolutely does. A one-size-fits-all anti-aging approach might be too simple.
Did they look beyond lifestyle interventions? What about genetics? Mice bred to age faster or slower?
Yes, they looked at those too. They examined mice with genetic mutations that cause accelerated aging progeroid mice, and also mice engineered to live longer, like certain dwarf mice that lack growth hormone signaling.
And what happened with their HSCs? Did they match the rest of the body's aging pace?
Remarkably, no. In the fast-aging mice, their HSCs were not prematurely aged compared to normal mice of the same chronological age, even though other parts of their bodies were falling apart.
Well,
And conversely, in the long-lived mice, their HSCs still aged pretty much on a normal schedule. They weren't significantly younger or protected, unlike other tissues in those same long-lived animals.
so the HSCs seem buffered, like they're running on their own schedule, regardless of the overall aging rate of the mouse?
Buffered is a great way to put it. It strongly suggests they have their own kind of independent aging program or clock. It's not tightly linked to the organism's overall aging trajectory, which is fascinating and, frankly, unexpected.
Okay, this is leading to the really crucial question then. If it's not the shared blood, not exercise, not calorie restriction, not even the overall genetic pace of aging, it really has to be something inside the cells, right? Or maybe their very specific location.
Exactly, and they did a really clever experiment to tease that apart. Remember the parabiosis, the shared circulation between young and old mice?
Yeah.
It turns out some of the old HSCs actually migrate and take up residence in the young mouse's bone marrow niche.
Oh, interesting. So you get old stem cells living in a perfectly young, healthy environment.
Precisely. The ideal test case,
Yeah.
right? Old cell, young neighborhood, young blood supply. If anything was going to rejuvenate them, this should be it.
The moment of truth. Did moving house make them young again?
Get this. Still no.
Yeah.
Even when these old HSCs physically relocated and lived inside the young bone marrow niche, bathed in young blood and surrounded by young support cells, they showed no changes in their aging markers.
Unbelievable.
Still high CD150, still poor engraftment, still the myeloid bias, still the replication stress signals.
So it's definitively not the environment. It's truly, deeply intrinsic to the old cell itself.
That's the major conclusion. The aging characteristics seem locked within the HSC, driven by internal processes that these external interventions just can't seem to override.
Which really brings us back to that idea of an internal clock, doesn't it? Like
Yeah.
the cell is just counting its divisions or tracking damage internally.
Exactly. It strongly supports that hypothesis. HSC aging seems primarily governed by something like replicative history or accumulated intrinsic damage, an internal timer, rather than being readily reversed by changing the external environment.
Is there anything that does change them? You mentioned transplantation earlier.
Ah, yes, there's a nuance there. The act of transplanting HSCs, taking them out and putting them back in that process itself, actually causes significant stress and lasting changes to the cell's epigenetics, how their genes are regulated. That happens to both young and old cells.
So transplantation complicates things.
It can complicate assessing rejuvenation because the transplant stress overlays everything. But, and this is important, even with that stress, if you transplant young HSCs even into an old, damaged environment, they can still function well and provide youthful blood production.
Ah, okay. So while you maybe can't easily reverse aging in old HSCs, you can potentially replace them with young ones that work.
Exactly. That offers a different kind of therapeutic avenue. It shows young cells have the robustness to function even in a less than ideal aged environment.
Okay, let's try and wrap this up. The big takeaway from this deep dive seems pretty clear, if surprising.
Mm-hmm.
Our blood stem cells, these critical HSCs, they seem remarkably stubborn, or refractory as the paper calls it.
Yeah.
They resist rejuvenation from things like young blood, exercise, calorie restriction, things that clearly help other parts of the body.
That's the core finding. And it highlights that this resistance seems to stem from deep-seated intrinsic factors within the aged cells themselves. They're playing by their own rules, essentially.
So thinking about the future of anti-aging,
Yeah.
this really forces us to ask, what does this mean if different systems in our body age so differently and need different kinds of help?
It really challenges that idea of finding a single magic bullet for aging, doesn't it? A one-size-fits-all systemic treatment might not work for every tissue, especially the blood system.
So what are the implications for therapies then? If you can't rejuvenate the old HSCs easily, what do you do?
Well, it suggests strategies might need to be much more targeted, perhaps directly hitting the specific molecular pathways that go wrong inside the old HSCs.
Or maybe intervention needs to happen way earlier, like focus on slowing HSC aging rather than trying to reverse it once it's established.
That's another possibility, yeah. Prevention rather than cure, perhaps. The study authors also suggest other ideas, like maybe finding ways to selectively remove the most damaged or dysfunctional old HSCs.
Clearing out the bad apples.
Kind of. Or maybe focusing on boosting the downstream cells, like the lymphoid progenitors, to improve immunity even if the main HSC pool remains aged.
Interesting. So lots of different angles potentially.
Definitely food for thought for researchers.
Okay, so here's a final thought to leave you, our listeners, with. If our blood system, the very core of our vitality in some ways, has this deeply ingrained internal clock for aging, what does that really tell us about the limits of trying to rejuvenate ourselves from the outside? How much control do we actually have over our cellular destiny?
That's the million-dollar question, isn't it? It suggests some aspects of aging might be more hardwired than we thought.